Hello, everyone, and thanks for listening to Medicenna's presentation on our technology. We call these evolutionary cytokines for developing revolutionary medicines. Let me explain it, but before I do that, I just want to make sure that to let you know that I will be making forward-looking statements, and therefore I encourage you to review our regulatory filings on CDR and SETI, etc. So let's talk about what we do at Medicenna. First and foremost, the focus of Medicenna is to develop, as I said, cytokines, and very specifically superkines. And these superkines are really focused on three different superkines, namely interleukin-2, interleukin-4, and interleukin-13. These different superkines enable us to modulate our immune system, and they are such that they can be used for treating dozens, potentially hundreds of different diseases, not only cancer. But I'll talk to you today mostly about oncology and cancer. And leading to that oncology program, the one that we have, which is the most advanced program, is called MDNA11. It's an interleukin-2 superagonist. This is a molecule that interleukin-2 has previously been approved. It's already a molecule that's in the marketplace to treat two different types of tumors, namely kidney cancer as well as skin cancer or melanoma. However, as I will explain to you later on, there are so many limitations and challenges associated with that molecule that we feel MDNA11 is potentially going to be the best-in-class IL-2 in development. The third molecule, or the program that I'll talk today about very briefly, though, is MDNA55. MDNA55 is a drug being used for treating patients with recurrent glioblastoma. It's by far the most aggressive, uniformly fatal kind of brain cancer. And we've got really exciting data and are ready for phase 3 clinical trials with a potential partner. And again, I explain to you that later on. And finally, really exciting that sort of new programs that are coming into Medicenna are based on what we call bifunctional superkines. And these bifunctional superkines, we call them, in short, biscuits. And these biscuits, although we won't have time to talk about those today, but we will certainly have an opportunity to provide multiple updates on this program at different conferences and so on and so forth. So let's get started first and foremost about the Superkine platform itself. This particular program, this particular platform, Medicenna and licensed under an exclusive arrangement with Stanford University. So we have global worldwide rights on all the IL-2, IL-4, IL-13 superkines. And the interesting thing here is that these superkines are such that we can fine-tune them to the way we want them to be used in the clinic for different patient populations with different types of diseases, whether they be cancer or even autoimmune disease or fibrosis or inflammation. So it provides us with so many opportunities to build out therapies for multiple disease types. And we will first and foremost today focus on just one particular type of interleukin or superkine, namely the IL-2 superkine, okay? And as I said to you earlier, the program that's most advanced is MDNA11. And this is perhaps really an important background slide on IL-2 in general. So as I said to you very earlier on, IL-2 has been approved for two different types of tumors, melanoma and kidney cancer. And this particular drug has been on the marketplace under the name Proleukin. It's been out there for about 20 years. And this particular molecule, as you can see on this particular slide on the left-hand side, what we show you is that it binds to a receptor known as the alpha receptor. And therefore, that binding to the alpha receptor engages our immune system in such a way that it actually stimulates immune cells that are going to protect the tumor. So for the drug to work, Proleukin has to be used in really high doses. And when you use it at high doses, it is able to stimulate some of the immune cells that will fight the cancer. The problem with this high dose is that it is incredibly toxic. Patients actually have to be admitted to intensive care unit for them to receive this drug because the side effects are life-threatening. And patients receive this drug over a period of 5 days, and they get these infusions every 8 hours. So you can imagine, although Proleukin has benefited a small subgroup of patients, the challenges are really huge, so that this particular drug has not really taken off. Our drug, though, as you can see on the right-hand side of this figure, is MDNA109. Now, MDNA109, it has been engineered, we call it an IL-2 superkine. As you can see from this image, is that it binds very specifically to the beta receptor, not the alpha receptor. And by virtue of that high affinity to the beta receptor, it actually stimulates your cancer-fighting immune cells preferably. And therefore, you don't have to use high doses. And therefore, as a result, you don't have to deal with the extreme toxicity, et cetera, that you generally see with Proleukin. So that aside, we have a number of other advantages. I'll talk about MDNA109 first, but MDNA109 eventually evolved into a molecule we called MDNA11. And the reason we did develop MDNA11 is simply that we simply took MDNA109 and increased the size so that the drug can stay in the bloodstream much longer. And that extended half-life of our drug allows our drug to be administered once every 2 weeks or once every 3 weeks in an outpatient setting, rather than Proleukin, which is required to be administered 3 times a day for 5 days continuously in an intensive care unit. So you can see the big divide and how beneficial MDNA109 and our MDNA11 program is. But before I go into generally talking about IL-2, let me give you a bit of background as to why IL-2 is so important. As you can see, over the past 4 years, there have been multiple transactions that have taken place with respect to IL-2. The very first one was in 2018 when Bristol Myers entered into a license agreement with Nectar Therapeutics that was developing an IL-2 that I'll describe briefly later on. And that was a $3.6 billion transaction. That program eventually failed in the phase 3 setting, and I'll explain to you later on why we feel that that particular drug failed. Subsequently, in 2019, just as Syntorex was starting their phase 1 clinical trial with their version of IL-2, they were then acquired for $2.5 billion by Sanofi. Again, that particular program is somehow similar to the Nectar molecule, but I'll explain why, again, we feel that the data so far from Sanofi do not show excitement, are probably, I would say, call them mediocre, and I'll explain to you why we think that's the case. The third program that you can see on the bottom left is Merck's acquisition of a company called Pandion. So Pandion was also developing an IL-2 program. It was in phase 1 clinical trials, not for treatment of cancer, but for autoimmune disease. And Medicenna has an earlier stage program, we call that MDNA209, where also this particular program, MDNA209, would benefit patients with autoimmune disease. By autoimmune disease, I mean arthritis, multiple sclerosis, et cetera. As you can see, Merck, again, paid quite a large amount, just under $2 billion to acquire Pandion. And then finally, more recently, just a month ago, we had Roche acquiring a program from a company called Good Therapeutics. This was a molecule which is a IL-2 linked to a checkpoint inhibitor, anti-PD-1. So this was a $250 million acquisition of a preclinical program. And again, we find that a really interesting space because we too at Medicenna have a similar bifunctional superkine in early discovery research phase. We call that molecule MDNA223. And this molecule, again, has our IL-2 superkine fused to a checkpoint inhibitor. And of course, each one of these, as you can see, different IL-2s have different uses and applications. But suffice it to say that the space in the IL-2 programs is really exciting. These are big pharma are certainly looking for the best potential IL-2 that is out there. So where do we stand and how do we compare MDNA11, which is our molecule, to all the others that are in development in the clinic at the moment? And I haven't put the data for Nectar here because it failed in a phase 3 clinical trial, but suffice it to say that Nectar's molecule was a molecule that had Proleukin, which is exactly the same drug that's been in the marketplace for 20 plus years. All they did was essentially take Proleukin and attach 6 molecules of plastic or polyethylene glycol to make the molecule larger so that it stays in the bloodstream much longer. But other than that, it did not do anything different. As you can see, Proleukin has a number of challenges, and clearly those challenges transfer to Proleukin, or rather Nectar's program. Sanofi does the same thing. They basically use the same Proleukin molecule, except that they have a better way of attaching the plastic. So we feel that Sanofi's molecule is potentially better than Nectar's molecule, but then again, it has the same backbone, namely the plastic backbone, to attach, and they still work with the same Proleukin. And then we have Alkermes. Alkermes is now entering phase 3 clinical trials. They've got a sort of decent data, much better data than Nectar's data. But again, they use the same Proleukin backbone for making their drug. And what they've done is, interestingly, they have avoided using pegylation or the plastic to increase the molecular weight and make the molecule a lot more selective. As you can see, it therefore does not have any of the liabilities that you normally have with attaching a plastic. Neoleukin is another company that is in the clinic. They started a few months before we did. Their approach, again, they use pegylated molecule. Their IL-2 is not actually an IL-2. They call it an IL-2, IL-15-like molecule, but they've essentially designed the molecule from scratch. It has no resemblance to interleukin-2 or Proleukin. It's a brand new molecule. So about 85% of the molecule has no resemblance to any human protein. The challenges there might be that that particular molecule could potentially be immunogenic. And if it is immunogenic, you have the issue of anti-drug antibodies, and therefore that might affect the potency of your drug. Werewolf is an interesting approach. This particular program is basically, again, taking Proleukin that's not changed at all from the original Proleukin, except that Werewolf, to make the drug safer, what they have done is they have masked the molecule, in a sense, sort of encompass the molecule in some kind of a shell, if you may, or sort of a mask. And that particular molecule essentially is inactive until it gets to the tumor. And once it gets to the tumor, the expectation is that certain enzymes like proteases might then unmask the molecule and then release the Proleukin right at the tumor site. The challenge, though, there is that Proleukin, as I said to you very early on, also stimulates Tregs, or regulatory T cells. They are immunosuppressive cells. And this is a challenge that we feel was also the problem with Nectar, because when all the pegs came off, what was left was Proleukin. And Proleukin was stimulating your Tregs, or immunosuppressive immune cells, dramatically. And that, again, was a challenge. So we feel that perhaps the approach being used by Werewolf may have sort of the benefit of localization, the benefit of reduced toxicity, but how that plays out in a human setting remains to be seen. And then we've got Xilio. Xilio, again, uses an approach which is essentially using a masking approach, but they've changed their proleukin in the sense that they've inserted some mutations. So it does not bind to Tregs and therefore will not stimulate your immunosuppressive immune cells. So perhaps Xilio has a molecule that is potentially providing the same benefits as werewolf in terms of the masking, but from an activity point of view, unlike MDNA11, MDNA11 is the only molecule that has been engineered so that it substantially stimulates your cancer-fighting immune cells, unlike all other molecules except for neoleukin. And then there's Superkine in the end, and the Superkine, again, they've engineered the IL-2 molecule, but at the same time, they're maintaining the activity so it binds to alpha, it stimulates Tregs, and we feel that there is potential for generating immunosuppression. And finally, of course, they rely on pegylation, which is associated with a host of other liabilities. So overall, if you look at MDNA11, it does not bind to alpha, therefore does not stimulate your Tregs. It binds exceptionally highly to your beta receptor, namely the cancer-fighting immune cells, and therefore that's what it does. The third thing is that it allows the drug to be administered once every 2 weeks or potentially even once every 3 weeks. So patients get this drug infrequently, which is great. Fourth is that it is the only molecule that you see on this list that has an albumin. And albumin is important because we know one thing about cancer is that cancer cells require albumin for the tumors to grow. So albumin allows MDNA11 to accumulate in the tumors very efficiently. We found, in fact, the drug stays in the tumor for 7 days or more. And therefore, I think that is really important, that differentiates us from everybody else. And finally, because we use albumin, we don't have to use pegylation to extend the half-life. And therefore, we don't have to deal with any of the liabilities of pegylation. So as you can see, I think I'm trying to convince you here clearly that we feel that MDNA11, amongst all the other programs that are in the clinic, is by far the best design and potentially the best in class. Okay, so let's look at some sort of data, or perhaps where we are with the program. We, of course, have seen very promising data in preclinical, in mouse models, in monkey studies, etc. We are already in the clinic. We are conducting this phase 1/2 clinical trial. We call this trial the Ability Study. And as you can see on the left-hand side, we are going through the sort of the first segment of the clinical trial, where we are enrolling patients. They are extremely sick. They have potentially as many as, allowing patients with 12 to 15 different tumor types to enroll in the study. The patients generally have failed anywhere up to 4 different lines of therapy. Most of these patients have undergone surgery, they've undergone radiation, plus 4 other therapies as well. And what we're trying to do here is, in this particular segment of the trial, is to optimize the dosing in these patients. So what we did was we started with the really low dose of 3 micrograms per kilogram, and then expanded or enrolled more patients into a second group known as the 10 micrograms. So these were the patients that received the low dose of the drug. And subsequently, when we found that the drug was safe, we increased the dose to 30 micrograms per kilogram, and then to 60 micrograms per kilogram. We've finished the first 4 cohorts and now proceeding and enrolling patients into the next higher dose cohort, which is the 90 micrograms, which is currently underway. So far, what we've done is we reported data from the first 4 cohorts. When we went into 60 micrograms per kilogram, we wanted to make sure, as you know, this particular molecule binds to the beta receptor 200 times more tightly than proleukin. So it has the potential of being extremely efficacious. So to avoid any potential toxicity issues, etc., what we did was we decided to use a different dosing regimen. We call it step-up dosing, where we administered the drug initially at 30 micrograms. We sort of habituated the patients to the lower dose at 30 micrograms for 2 cycles, and then increase the dose to 60. This is what we're doing now. And we're doing the same thing at the 90 micrograms, is treating patients at 30 for 2 weeks and repeating that process again and subsequently going to the 90 microgram dose. So here, what we are being able to accomplish, the idea here is to get to as high a dose as we possibly can to get the best outcome for the patient without creating safety liabilities. But at the same time, use a dose at 30 micrograms where we know that the molecule and the patient is potentially benefiting. We've already seen, for example, at the 10 micrograms per kilogram, we've got a patient now on the study for about a year now, and that patient continues to show tumor control very effectively in this particular 10 microgram dose. So we feel that at 30 micrograms, we've seen that we stimulate the cancer-fighting immune cells by about 100%. So even though the patients are getting the 30 microgram dose, we feel that the drug is benefiting the patient. And then we are boosting it to 60 or 90. And who knows, we might even go to a higher dose of, let's say, even 120 or 150, depending on what the data on the 90 microgram show. So once that is completed, we are planning, hopefully by early next year, to proceed to the next phase of the trial, which we call the dose expansion, where patients with 3 or 4 different tumor types, about 40 patients will be enrolled there, where we will administer what we call the recommended phase 2 dose. The recommended phase 2 dose will be established in the first portion of the trial. And that will be used to treat patients with, hopefully, these patients will be the type of patients that are not really end-stage patients, but these patients will have a much more healthier immune system. These patients will be selected in such a way that they are likely to be the patients that will eventually be used in a commercial setting, so that the phase 2 data will help drive the phase 3 design and eventually into a registration trial for this particular molecule. And then finally, we've seen preclinically, but also we've seen others demonstrate that when you combine IL-2 with other immunotherapy, like a checkpoint inhibitor, then there is a synergistic effect. And as you know, checkpoint inhibitors like Keytruda, which is the drug that we're really combining MDNA11 with, for which we've entered into a clinical collaboration with Merck, who will be supplying us this drug, working together with us in terms of the design of the combination part of the trial. That would be very useful for us to see what can we do to those patients with patients that have cancers that have failed therapy with Keytruda. Can we improve their outcomes if we combine them with MDNA11? Remember, Keytruda is perhaps the most successful immuno-oncology drug out there in the marketplace, with sales approaching about 20 billion or so. And that particular therapy itself, even though it is the most successful drug, still only treats and benefits about 25 to 30% of the patients. The remaining 75 to 80% of the patients don't benefit from Keytruda. And if we can increase the benefit rate from 25% to 50% by combining with MDNA11, that would be really fantastic. So that's the plan. We will plan to start the combination part of the trial middle of next year. And together we'll have lots of data coming throughout, from starting Q4 of this year all the way into Q3 and Q4 next year as well. So we'll have regular data coming through by virtue of these 3 different cohorts that we are treating. So going on to the next slide, just to give you an idea as to what the data look like, here, what we're measuring here is lymphocytes. These are the population of your immune cells that generally would need to be stimulated. These patients, as you can see, particularly those patients at the 10 and 30 micrograms, had very poor baseline immune status. They had very low lymphocytes. And you can see that on the right-hand side, with 10 micrograms and particularly at 30 micrograms per kilogram, we are nearly more than doubling the population of your lymphocytes. And then at 60 micrograms, again, you see a further increase in the population of your lymphocytes. So that's really important, particularly because once you are able to stimulate your lymphocytes, hopefully you've stimulated your cancer-fighting immune cells, and these are the ones that will eventually fight cancer for you. Going to the next slide, we are also, one of the problems with Proleukin, the drug that's been approved and some other companies continue to use, is that it does cause sort of a side effect. We call that eosinophilia. And associated with this eosinophilia is vascular leak syndrome. Now, what does vascular leak syndrome mean? It actually means that your blood vessels start leaking, and when your blood vessels start leaking, your blood pressure drops very dramatically, and you could have a life-threatening event. And therefore, that's why Proleukin is administered in an intensive care unit to prevent patients from potentially dying of vascular leak syndrome. You can see that from these data here, irrespective of 10, 30, or 60 micrograms per kilogram, the effect of MDNA11 on eosinophilia is negligible. And therefore, we're not seeing the huge increase to demonstrate that this drug causes potentially vascular leak syndrome or potentially sort of life-threatening side effects. So that is really encouraging. Next, of course, is, how have patients done with MDNA11 so far? So this is what we call a swimmer's plot. And on the left-hand side, it shows you the different doses that have been administered to patients with different types of tumors. We've got patients with melanoma, sarcomas, with pancreatic cancer, with head and neck cancer, kidney cancer, and so on and so forth. So these patients, just to give you a bit of background, these patients, as I said, have end-stage disease. 80% of these patients also failed checkpoint inhibitor therapy, such as Keytruda, Opdivo, etc. So you've got these patients who have failed chemotherapy, have failed immunotherapy, etc., and entered the clinical trial. As you can see here, out of the 14 patients that we have data on, 5 of those patients had tumor control, and one of them, particularly interesting, is a patient with pancreatic cancer. This particular patient was initially at 12 weeks determined to have stable disease, meaning the tumor had stopped growing. By week 16, the tumor had shrunk by more than 30%. And more recently, we've confirmed that this particular patient that we call a unconfirmed partial response subsequently had a confirmed partial response. And this is significant because pancreatic cancer is by far the most aggressive type of tumor. It is really aggressive. It's very difficult to diagnose these patients. Even the patient in this particular clinical trial already had surgery. So the primary tumor in the pancreas was removed. This patient then received chemotherapy. This is namely a combination of 4 different chemotherapy agents. The patient tumor continued to grow and metastasize to the liver. The patient then received Keytruda. Again, these patients continue to have the tumor continue to grow. But what was also interesting is that this patient was not able to tolerate Keytruda. This patient also received, before receiving Keytruda, apparently also received a combination of another drug, which is Abraxane and gemcitabine. So you can see that this patient has undergone multiple therapies and then finally entered into the ability study with our drug MDNA11. And what is really exciting to see in this patient is the patient is still on study, is receiving the 40 microgram per kilogram dose, or rather the 60 micrograms per kilogram dose. And you can see the patient has a partial response that is confirmed, and the partial response continues to deepen, meaning the tumor is continuing to shrink even more than what it was 16 weeks ago. The other interesting patient is the patient at the 10 microgram per kilogram dose. This is the patient, again, that failed 3 to 4 different lines of therapy, including checkpoint inhibitor therapy. And as you can see, even at the 10 microgram, this patient's tumor stopped growing. And this patient has subsequently received a higher dose of 30 micrograms. And more recently, just last month, started to get the higher dose of 60 micrograms per kilogram. The tumor continues to be stable, and therefore really encouraging that this patient now is nearly on the study for just about a year without having to deal with toxicities, et cetera, and being able to tolerate this drug quite well. So really exciting data, considering that we've got an end-stage patient population, promising when we look at other therapies out there or other IL-2s. I would say that the effect we've seen in pancreatic cancer is really unique because this is a difficult tumor to treat. We know Proleukin, for instance, does not benefit patients with pancreatic cancer. We've seen the other data from our competitors is that none of them have shown single agent activity of the IL-2 in pancreatic cancer patients. So hopefully this provides us some confidence that we probably have the best-in-class molecule as we advance the program into higher doses and eventually at the dose expansion phase of the study. So I will transition now to talk briefly about MDNA55. This is our drug for recurrent glioblastoma. Again, a really exciting program. This is a different drug. It's not an IL-2 drug, but it's an interleukin-4 drug. So what we've done is we've taken interleukin-4, attached it to a very potent toxin, and this drug is delivered directly in the tumor. It's one-time administration into the tumor. And what we see is that with one treatment in these patients, on the right-hand side, what you see is the survival of patients with recurrent glioblastoma. Again, these were end-stage patients, and we see the median survival there is 15.7 months, which is quite substantial considering that everything in the marketplace today that have been in use for the past 20 years provide median survival in the 6 to 7 month or 6 to 9 month range. And for us, what we did was we said, okay, let us try and do a separate study where we enroll patients that had received other approved therapies that met the same inclusion-exclusion criteria as our clinical trial. They had the same profile. We matched these patients using 11 different prognostic factors like tumor size, tumor location, age of the patient, their biomarkers, etc. Once we matched it and we unblinded that particular group, we realized that the patients that receive other therapies had a median survival of just over 7 months. So really exciting to see that MDNA55 with a single administration is more than doubling the survival of these patients. And therefore, we find this is exciting. We're looking forward to partnering this particular asset. So we are at a point we are phase 3 ready. We've got to go ahead from the FDA in terms of conducting the clinical trial. But the key thing here is to find a partner who's going to eventually commercialize this drug for this huge unmet need indication. So that's where we are with our drug, and the key milestones obviously coming up are first and foremost that we have just reported this just last month, the data from the cohort 4, where we found that we had now 5 out of 14 patients benefiting from the drug, including a confirmed partial response. We are enrolling patients in the 5th cohort. We'll have initial pharmacokinetics, pharmacodynamic data from this group before the end of the year. But more importantly, in late January, early February, we'll have scans from these patients. So we'll know whether the 60 micrograms or the 90 micrograms is doing better at the 90 microgram dose or not. And then after that is completed, once we've established the recommended phase 2 dose, the plan is then to proceed with a dose expansion middle of next year, and also the combination portion of the study, which will have us a bunch of data at the end of 2023. So we'll have more data from the dose expansion monotherapy at the middle of this year. We plan to start in the first quarter next year. Middle of next year, we will have the scans, at least the first 1 or 2 scans in the majority of patients. And then at the end of the year, we'll have data from the combination part of the study. So we have, as you are aware, that we recently raised $20 million. That fund, plus our existing cash balance of about 19 million Canadian, allows us to achieve each of these milestones that I show you on the left, and also takes the cash into 2024, plus also allows us to get a program, which is our BiSKIT program that I haven't had a chance to talk to you about, but that will allow us to take that program into IND-enabling studies. So overall, as I said, we have cash of 19 plus 20 million US that was just raised. We have no debt, no preferred shares, really clean structure. We have common shares across the board. one class, one type of common shares, there's 70 million of them, and then on a fully diluted basis, 92 million shares. So I will stop here and basically convey to you that we feel that we've got exciting IL-2, IL-4 programs, and also potentially other earlier stage programs that we call biscuits. And clearly over the next 2 to 3 quarters, we expect lots of news flow and hope to provide you with updates every quarter going forward. Thank you.